Hello, hi, a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. So welcome to my lecture. So today we are in lecture 5 of the topic molecular basis of inheritance. Today let's try to cover translation, and let me give you an introduction of what is Human Genome Project, that is HGP. Alright, so this is the agenda for today's lecture. So are you ready to move forward? So we have recently completed transcription. So before that, we spoke about replication. So I hope replication and transcription are pretty clear. So today we're going to talk about the process of translation. Yes, any doubts till this? Hi, and okay, so this is ASM. I'm sorry. Yes, hello, hi, a very happy good evening. So thank you so much for joining my lecture today. So please do like, share and subscribe for all those who are watching my lecture. So I thank you in abundance, and please don't forget to like, share and subscribe. Alright, so shall we move into today's lecture? So shall we talk about the process of translation? So translation is a part of central dogma of life, which started with DNA replication. Then we went on to talk about the process of transcription, and today it is translation. Alright, so shall we move forward? So here we go. So before we go and talk further or dwell into this topic of translation, so I have an announcement for you. So this evening we are meeting once again, but not for any lecture, but for a very interesting and motivational session. So today at 6 p.m, don't forget to join me. So we are going to talk about something really interesting, something very different. So just sit back and listen to the stories, so which says good things take time to happen. Okay, so this is a kind of motivational session because you guys very frequently drop out of motivation. Yes, so very soon you guys get frustrated, very soon you guys reach the top temperature. So yes, so now and then your motivational session is necessary. So please join me at 6 p.m. So we're going to talk about how good things take time, and how to stay motivated and how to calmly prepare. So the more you panic, the more you tend to fail. So please relax and sit back. So please join me at 6 p.m. and listen to the stories that I have to tell you. So if you have any siblings who are also in that same track, who's getting depressed, who's getting frustrated because of the online classes and work from home mode, so please bring them here. So this session will give you a lot of energy. Alright, yes. Hello, hi, Anthony Abi. A very happy good evening. Yes, I'm good, thank you. How are you? Yes, you're waiting. Okay, I'm eagerly waiting for this interesting session with you guys. Alright, so please do like, share this session with your friends and family. Okay? Okay, so moving forward, so I hope you guys have decided something about the crash course, because time is ticking off and the price will also go back to its original price. You will not have this offer very soon. So please, it is 1,999. You have 3 subjects, that is your P, C and B. So every day 1 hour you have the lecture, and Saturdays you have doubt counters, and Sunday you have unit test, and I am handling biology. So as for biology, I can guarantee you it's going to be a very interesting session. Alright, yes, so please give a thought about it. Yes, Darni, a very happy good evening. So thank you for joining my lecture today. So please do like, share and subscribe. Alright, so shall we talk about the process of translation? It is pretty short. So once I am done with translation, almost the major portion of this chapter is done. So what is the next most important part that is remaining in this chapter? It will be a regulation of gene expression. So we will talk about lac operon. So this week we'll be done with molecular basis of inheritance from class 12, and from class 11 we'll be done with breathing exchange of gas. We'll also be done with body fluids and circulation. So this week we'll be done with 3 chapters. So 6 days, 3 chapters is a huge thing. So please pay attention, because your exams are nearing. So we do not know the exact date, but yes, we all can anticipate this, that your exam is nearing, right? Ma'am, please, after physiology, can you start plant physiology? Yes. So after human physiology, it is plant physiology. But if you want it very instantly, so I have taught plant physiology 1 round on YouTube. So you can go back and find the videos, or else you can wait, because now we are in body fluids. So after which we have 4 more chapters to go into plant physiology. But if you have any doubts, if you want to know a concept very soon, so please look into those lectures. And if you have any doubts, so beneath the description box of my video, there is a Telegram link. So you can join into the Telegram link and get to me, so that we can clarify your doubts. Alright, ASM? Yes? Okay. So we were talking about the process of DNA replication. So then we went on to talk about transcription. So we spoke about transcription unit and the gene. So the three parts, right? So then types of RNA and the process of transcription. So today we will talk about what is your genetic code. Yes, so why do we need your genetic code? Mutations and genetic code, how are they related? What is tRNA molecule? So how tRNA is playing a role here. And then finally we talk about the process of translation. So let's see how a translation process is different from transcription and replication. Actually, when we started talking about your transcription, we felt a bit different because in replication, both the strands of DNA replicated. But in your transcription, only one strand was copied. But in translation, what is the difference? So here we see, welcome to the process of translation. So we were talking about your central dogma of life, if you remember. Hello, hi Shimpi, a very happy good evening. How are you? Your cold is fine? Okay, Shimpi, I'm good, thank you. So please like, share and subscribe. Okay. So I hope you remember central dogma of life. So central dogma of life was stated by Francis Crick. So he is the scientist who spoke about central dogma of life. So he said that DNA replicates during the S phase of the cell cycle, and then the information is copied onto the RNA. This process is transcription, and then the transcription is decoded as proteins, that is translation. Alright, okay, so we are not going to talk about a recap of your replication, but we'll talk about the recap of your transcription process. I'm just checking if my slides are right. Okay, yes. Okay, so I hope you remember the process of replication. So no, your cold is still there. Oh, I'm really sorry. Ma'am, can you find plant physiology in the playlist? Okay, so please ping me in the Telegram. I will share the link of plant physiology with you. All right, yes? Okay. Okay, so replication, I hope you remember the main set of enzymes. So now let's talk about the process of transcription. So in transcription, only one strand is copied, and I hope you remember both the reasons why only one strand is copied. So which strand is copied? If this is your DNA, so this strand acts as your template strand. So this is your template strand, and this is also known as your non-coding strand. And this is your coding strand by weird terms. Right? So information from only this strand will be copied because your RNA polymerase, which is a very important player here, can polymerize only from the 5' to the 3' direction. Alright, so here the next part that you have to remember. So here you have a promoter, so here you have a terminator. So in between this is your structural gene, and these three make up your transcription unit, right? I hope you remember this. Yes? Okay, so now the next part that you have to remember in the process of transcription is that, yes, hello Munna, hi. So how nice of you. So you are here for my 4 o'clock session as well. You turn up for my session at 6 as well. So very good, Munna. Thank you so much. Yes, I'm good. So please do like, share and subscribe. So Munna, I have a motivational session today at 6. So please join me. I'm gonna tell a lot of stories. So please bring your friends as well. Okay, Munna? Yes? Okay, yes, Antonia B, it's 5' to 3', you're right. So further, what you should remember about the process of transcription is, so this is clear, so this is the first part. So wherever there is promoter, so this is your upstream and this is your downstream. Okay, so then moving further, so we spoke about translation in your eukaryotes as well as prokaryotes. So I guess you all know that replication happens in the nucleus because that happens during the cell cycle, and then and transcription happens in the nucleus. But this is in case of eukaryotes. But in prokaryotes, since we do not have a well-defined nucleus, everything happens pretty nearby, right? And your prokaryotes have a lot of cistrons accumulated together. So your prokaryotes are polycistronic. So this is prokaryotes. Okay, so what is a cistron? So cistron is a piece of gene that codes for a protein specifically. So gene can be one which can code for protein, mRNA and tRNA. And now when it codes especially for your protein, it is known as a cistron. So in your prokaryotes, the cistrons are clustered together. So it is polycistronic. Whereas in your eukaryotes, your cistrons are monocistrons. Okay, so how does the process of transcription in prokaryotes happen? So transcription in prokaryotes happen by 3 steps. One is initiation, elongation, and then you have termination. Okay, so initiation is aided by the sigma factor of RNA polymerase. So RNA polymerase binds to your promoter. So in prokaryotes, it binds to Prunau box. Right, so in prokaryotes, it attaches to your Tata box. So this is the recognition site of your RNA polymerase on your promoter. Okay, and here it elongates, so you have your mRNA being formed, and then termination is where you have your Rho factor. So your Rho factor helps in termination. So the mRNA that comes out of this process immediately moves into the process of translation. Alright, yes, initiation, elongation, termination. Yes, Antonia B, absolutely right. Okay, yes. So Munna, you're disturbing Shimpi? Huh? What is Tata box? Okay, so your RNA polymerase will bind to the promoter and initiate the process of transcription, right? So where exactly your RNA polymerase will bind in the promoter is in a sequence known as Tata box. The sequence is TAA box. Okay, so that is the sequence which your RNA polymerase will recognize on your promoter. Okay, Shimpi? Yes? Yes? Okay, yes, so Munna is giving you advice. Yes, Swami, a very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. How does eukaryotes enable the process of transcription? So here also you have 3 processes. You have initiation, you have elongation, and you have termination. So you have initiation factors which play a role. So you have elongation factors which play a role, and finally you have termination factors which play a role. Alright, but in eukaryote, the mRNA that comes out here after these 3 processes, so this mRNA is an immature mRNA. So this mRNA is known as primary transcript because it is the product of transcription. It is known as primary transcript, or it is an immature mRNA. Okay, but this mRNA has both exons as well as introns. Right, so these are introns. So what is the first step that happens? The mRNA is immediately capped. Right, so the cap is done by cap is done at the 5' end, and it is done by methyl guanosine triphosphate. So this caps your mRNA so that it does not undergo degradation, right? And then further at the 3' end, this is the 3' end. So after capping, so your mRNA undergoes a process of splicing. So splicing is a process where your introns are chopped off. Introns are intertwining sequences, so they are chopped off by the process of splicing. Now the last step is tailing. So as soon as the splicing is done, so you have nearly 200 adenine residues getting added at the 3' end. So we call this as poly A tailing. So this ensures that the mRNA is mature enough and it is ready for the process of translation. So this is the process of transcription. Alright, is this clear? Yes? So here you have to remember you have 3 types of RNA polymerase in your eukaryotes. In your eukaryotes you have 3 types of RNA polymerase. So we call them as RNA pol 1, RNA pol 2 and RNA pol 3. So what is the function of each of these polymerases? So RNA pol 1 is used to transcribe your rRNA and this is your tRNA. Your RNA pol 2 is used to categorize heteronuclear RNA, so which is nothing but your mRNA. So here for our process of transcription, we use your RNA pol 2. Clear? Yes? Okay. So now we enter into the process of translation. So now let me give you a small introduction to this process of translation. So I have my mRNA here, right? So this is my 5', this is my 3'. So this is my mature mRNA. So here I have poly A tail. So here I have a cap. This is guanosine methane phosphate cap. Okay. Now if this has to transcribed, so now if this has to be translated, it is going to produce proteins. So proteins are polymers of amino acids, right? So I am going to have something like this. Right, so this is what I am trying to prove at the end. This is an mRNA, so at the end it is an RNA, so it has nucleotides here. Yes, I agree, it has exons. So exons are nothing but nucleotide sequences, right? So from nucleotides, it has to be converted into a polymer of amino acid. How is this possible? So from DNA, if the information has to be copied on to the mRNA, it is still easy because your DNA as well as your RNA both are nucleotides, right? So it is pretty easy. But now I have to change my base itself. The basic platform itself changes. From nucleotides, I have to produce amino acids. So how am I going to do it? So how did scientists come to a conclusion that yes, translation happens like this? So it was a major challenge, okay? So scientists did not know how it is happening. So it took a very, very long time. So it took a lot of scientists from so many fields to come together to attempt to how this will happen. So how did they finally do? So for this discovery, yes, so you had nearly so many scientists. You had a physicist, you had a chemist, you had a biologist, you had a genist, you had so many people. And among this, we had one Indian, and it is very prestigious to name him. His name was Har Gobind Khurana. So he was also a part of this discovery, the discovery that I'm going to talk about now. So among so many foreigners, he was one Indian who was a part of this discovery. And even today, on his name, so we Indian students are given scholarship to go abroad and work in the field of genetics. So that is known as Har Gobind Khurana scholarship. So that is still valid. So even today it is happening. It is a very prestigious scholarship and it is only for your life science students. So what is this discovery? Okay, so you have nucleotides. So nucleotides are made up of 4 nitrogenous bases, right? So the 4 nitrogenous bases are adenosine, uracil, guanine, cytosine, because it is RNA, right? And here it's amino acids. So totally we have 20 amino acids. So 4 nitrogenous bases has to convert into 20 amino acids. So how is this possible? Suppose if I go to a country which I am very new to and I don't even know the language of the country. Say for example, I am going to Mexico. In Mexico, Mexican is spoken. English is very, very rarely spoken, okay? So those people are pretty fluent in their native language, so English is a rare term for them. So if I go to Mexico, okay, so I do not know the Mexican language, but I go and land there. So I need a translator, right? I need someone who can connect me with the people of Mexico, right? At least if I land in the airport, someone with my name placard, I should be able to read the name on that placard, right? I should be at least able to translate and read, okay, so this is, my name is written on the placard, so I should go to that person. So this is the person whom I should meet. At least that translation should be there, right? So that is what we are trying to find out here. So they found out that we have 4 nitrogenous bases and we need 20 amino acids, right? So we have 4 nitrogenous bases and we need 20 amino acids. Then in what way the nucleotides are read? So that was their question. So they did permutation and combination. So 4 power 1. If each codon can give us 1 amino acid, it will only give us 4 amino acids. So this is not possible. Because every A codes for proline, every U codes for phenylalanine, then it is not possible, right? So then they did 4 square. So 4 square is 8. But 8 is not our target, right? Our target is 20 amino acid. So the code cannot be 2-letter codons. So it cannot be that every 2 nucleotides form a code, right? So that is also not there. So then they did 4 power 3 and they found out 64. Then they found out, yes, you have 64 messages which code for 20 amino acids, and each message is in the form of a 3-letter codon like this. So every 3-letter form the code. Okay, so we call this as codon, AUG. So this is a 3-letter code and this coded for 1 amino acid. So whenever this sequence is read on the mRNA, and we understand this codes for the amino acid methionine and this codes for the amino acid phenylalanine. This codes for the amino acid proline. So we understand like this, and hence the genetic code was born. So this is known as genetic code. So this was a groundbreaking discovery in the field of genetics, alright. So codon trick, because you want to remember the entire codon. Even I don't remember the entire codon. It is quick to understand and do the coding, right? Yes, let me teach you how it happens. So this is the introduction to translation. There is no complementary here between the nucleotides, because one is nucleotides, the other one is amino acids, right? So the power or the rule or principle of complementary does not work here properly. Thus the genetic code was proposed. It was 4 power 3 is 64, and this conclusion was given by George Gamow. Though you had a lot of scientists who worked hard, so he was the fellow who came out with this permutation and combination. So here we have the majestic genetic code. So I told you, no, so every 3-letter codes for 1 amino acid. So which means you should have 64 messages. You should have 64 3-letter codons. So what does that mean, or how do you find? Okay, so let me tell you how to do it. So we have 4 bases. So we take the basis of mRNA. So U, A, G, C. So 4 bases, right? So I write U, A, G, C. Done? So I write the same one. Okay, so it can be now U, A, G and C. So understand? So it is U, U, U. So then it will be U, A, U. This is U, G, U, U, C, U. Okay, so this is how you do the coding. So how? U, A, U again. So this is U, A, sorry. This is A, so this is A, U, U. So this is A, U. then it will be AGU, right? So this is how you do the coding. Okay, so like this you finish off the entire table. So see, so this is just an example I gave. So I have the first codon, the second codon and the third codon. So it is UUU, right? Then it is UUC, UUA and UUG, right? So please do the coding in this order and remember the order of amino acids. So in a way you can remember the table if you want, but as such the entire table is not needed for you. It is for much higher level. Okay, so you do not have to remember the table, but you should know how to arrive at the genetic code. So you write the first base 1, 2, 3, 4. 1, 2, 3, 4. You write it like this. Then the third base you fit all the 4 into one column. So then you do the multiplication. So please go in this order: UCAG, UCAG, UCAG. So then if you go in this order, you just have to remember, if you have how many columns here? 4 columns. So remember, like the first column 2 are phenylalanine, next 5 are leucine, then isoleucine, 1 methionine and then valine. So you can remember this if you want, but otherwise this is how the codon is generated. So Shimpi, did you understand how the genetic code is being made? Yes? So should I solve it for you? Okay. U, C, A, G. I write only U. Okay. So here I have U, C, A, G. So now it will be U, 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 C, U, U, A, U, U, G. Right. So here it will be C, U, U, C, U, C, C, U, A, C, U, G. Right? So now you're getting? Yes, so you do not have to mug up, but this is how you do it quickly. So similarly, write all the bases and do the crossing. You get the 64 codons. Okay? Yes, so this is how I remembered for an exam. So I still remember because I couldn't remember everything. So suddenly it said CCA is proline, then CAA is glutamine. So I couldn't remember, but I know how to do the crossing and I was pretty fast in writing. So when they gave me the question paper, I saw the question which was related to genetic code. So fatafat, I took out the first page, I drew the genetic code. I know in which order the amino acids come, so I filled it out, and then it was like a dictionary reference. For each question, I happily looked into the genetic code and I got all of it right. So this is one easy way if you can remember. So remember this tabular column alone. So this is pretty easy. Serine, proline, threonine, alanine, right? So it is SPTA. Right, so you can remember it as SAT-P. So whichever way you can remember it. So this is a very effective way. But now pay attention, we have 64 codons here, but there are some codons which says stop. So you have 3 codons which stays stop, which means these codons do not code for any amino acids, but they are stop codons. So when you have this sequence in your mRNA, the process of translation stops. When you have this sequence in your mRNA, the process of translation stops. So out of 64 codons, only 61 codes for amino acid and the remaining 3 codes for stop codon. So please remember the codes for the stop codon: UAA, UAG and UGA. So these 3 are stop codons which I want you to remember. Alright, so since this was a groundbreaking discovery, you have some features of your genetic code. So what are the features of your genetic code? Your code is a triplet. Yes, the message is in 3-letter form. So it is a triplet. So totally you have 61 codons which code for amino acid and the remaining 3 codons code for stop codons, right? So that is how it is, because you have terminator in your transcription, right? But here we do not have a terminator. So here we have only stop codons. So how will you know the process has to stop? Anywhere it has to stop, right? So the stop indication is given by a stop codon. Alright, Shimpi? Yes? So some amino acids are coded by more than one codon. So did you see your valine, your alanine was coded by more than one codon? You had so many 3-letter codons for a single amino acid. So this feature of the genetic code is known as degenerate, when you have more than one codon for an amino acid. So codons are read on the mRNA in a contiguous fashion. It has no punctuations. You can read it continuously. It has no punctuations, right? Then the code is universal. It is pretty common from bacteria to humans. So everywhere AUG codes for methionine. So be it bacteria, be it elephant, be it Antony, be it Shrimpy, everywhere AUG codes for the amino acid methionine, and everywhere UUU codes for phenylalanine. So everywhere CCC codes for proline. So this genetic code is universal. So AUG is the start codon and it codes for methionine. Alright. So this is also known as your initiator codon. So your UAA, UAG and UGA are stop codons. You should remember this. So these are the features of your genetic code. Okay, but how did people find this out? How did they find this out? How did they find out? Okay, so we assume it is the genetic code, but we have to cross check it, right? If that is how it is happening inside the body. This is a hypothesis. Initially, your genetic code was an hypothesis, but then it was proved. It was actually seen this is how your cell does translation. But how did you think they tried this? How did you think they tried this? See, my question is, are you able to think? Because I just want you to think. I don't want you to answer anything. So this is AUG, GUC, say GAG, UUU. So we found out it is triplet codon, right? So this is from 5' to 3'. So someone has to read this codon and then bring the amino acid, right? So amino acid is synthesized by your cell. We have essential amino acid and non-essential amino acid. So amino acid synthesis happens inside your cell. But who exactly reads this and translates this? How does it happen? Who is the mediator here? Who brings the methionine here? Who reads that AUG codes for methionine? Who knows this? Which part of the cell? Yes, just like I told you, if I land in China and if there is a person holding my placard in the Chinese language, so how can I read it as Krishna? I need someone who can help me in this transition, right? So that transistor or your adapter molecule is your tRNA. So tRNA helps communicate this translation. So what is an adapter? So when you're going to charge your phone, you only do not need your USB cable. You need an adapter as well, to which you can plug your USB cable and charge, right? So adapter is something which connects to different things, right? So similarly, your tRNA is the adapter. Your tRNA can read AUG codes for methionine and it brings the methionine and attaches here. So that is how the process happens. Alright, so now let's talk about the structure of tRNA, because tRNA is a very, very important player here. So let's see. Okay, so before we go into tRNA, you have mutations and genetic code. So what is mutation? So effects of large deletions and rearrangements in the segment of DNA is easy to comprehend. It may result in the loss or gain of a function. So what is this? So here if I write, so let me change the color. So if I write the hat was red. So this is the sentence, alright. So in this sentence, every word has 3 letters. So assume this is our codons. So it is a triplet codon and it codes for an amino acid. Just assume, okay? So every letter, every word in this sentence has 3 letters. Assume this to be your codon. Assume this codes for an amino acid, this for an amino acid, this and this. This is how my reading frame is, right? So the hat was red. This is my reading frame. Suppose by the process of mutation is the sudden change in your DNA. So now by the process of mutation, if my one letter changes, if I delete off A, because of mutation, A is deleted, then what will happen? How will the reading frame be? So it will be THE, then it will be HT, yes, W, ASR, ED. So now your reading frame changes. So this made sense to you. You were easily able to read the hat was red. But now you cannot read this, you can only read this, right? So even if there is a change or deletion in one amino acid, so it leads to mutation, or this leads to the change in your reading frame, right? So this is what I mean. So here in sickle cell anemia, it changes from GAG to GUG. So here the reading frame changes, but here you have a substitution. Even then this changes because this codes for a different amino acid, this codes for a different amino acid. So that is why your genetic code is related to the topic mutations. Alright, clear? Okay. So with this, it's clear. I hope I have explained what is reading frame. So now talking about your transferred RNA, that is your tRNA, which acts as an adapter molecule. So Francis Crick found a mechanism, or he insisted that there should be a mechanism which can read the code and link it with amino acids. So the Chinese placard has the name Krishna, but I don't know how to read. But I have a translator who will look into the board and read Chinese, as well as he can translate that to me in English, right? So is the function of your tRNA molecule. Amino acids have no structural specialities to read the code uniquely. Amino acid itself cannot read its code and join and form a polymer of proteins. No. So we need a connector here. The connector here is your tRNA. Alright, so postulated the presence of an adapter molecule. So Francis Crick only postulated the presence of an adapter molecule. So he said that this reads the code and it has one hand to bind with the amino acid. So these molecules are called as transferred RNA, which were then called as soluble RNA. So today we call it as tRNA. So T stands for transferred RNA, but way before it was called as sRNA because it is also known as soluble RNA. So soluble RNA or sRNA is the same thing. Alright, so here we go into the structure of tRNA. So this is your tRNA structure. Okay, so this is your amino acid end of your tRNA. Okay, so this is the structure of your tRNA. So this is your clover leaf structure. Fine? Yes? Okay. So now this is the amino acid acceptor end. So the amino acid will come and bind to the tRNA. So imagine this to be a translator. So this reads the Chinese language. It also tells me in English. So here it attaches the amino acid. Depending on the code, it brings an amino acid here. So this is the anticodon arm of your tRNA. This is the amino acid code, and this is known as your pseudouridine. You can say it as 5' arm. Okay, it is pseudouridine arm. Okay. So this arm helps in the attachment of tRNA to ribosomes. So during the process of translation, ribosomes are important players, right? So your tRNA binds inside the ribosome. So this arm helps in the attachment of the ribosomes. Alright, so the tRNA has an anticodon loop, that is this, okay, the loop, the bases which are complementary to the code. So it will be like this. So I have my mRNA molecule like this. So I have the code on the mRNA. So the tRNA produces the anticodon, the complementary codons. The amino acid acceptor arm binds to the amino acids. So tRNA is specific for each amino acid. For initiation, you have another specific tRNA which is known as initiated tRNA. So there are no tRNAs for stop codon. Whenever your ribosomes read the stop codon, it will stop the process of translation. Okay, so this is how it is. So this is your mRNA. So this green one is your ribosomes, the larger subunit, the smaller subunit. Okay, and these are your tRNAs. Can you see? So tRNA reads this code, it produces the complementary code, and this complementary code is quickly comprehended here and it brings the amino acid, right? Is it clear? So the process of polymerization of amino acids to form polypeptide. So the order and sequence of amino acids is defined by the sequence or codons in your mRNA. So amino acids are joined by peptide bond which requires energy. So in the first process of translation, so translation also has three processes: initiation, elongation and termination. Alright, so how does this process of initiation happen? First, the tRNA has to be active. So first the amino acids will attach to the tRNA. For that, the amino acids have to be activated. Suppose if you are going out, you get dressed, right? So now if your amino acid is attaching to the tRNA, it will get activated and only then it will bind to the tRNA. Now the tRNA is activated. It is ready to enter inside the process of translation. Okay. So activation of tRNA. Okay, so this is how it is. So you have three sites in your ribosomes. So now your tRNA is bringing an amino acid and it puts it here and it will join it with the polypeptide and go. So each amino acid, each tRNA molecule carries an amino acid, right? So amino acid attaches to the tRNA and this is your charged tRNA. Can you see? This is how it happens. So this is the incoming tRNA. So it binds to the polypeptide and it exits. So you have your P site, A site and E site. So this is the incoming tRNA and it moves here. So it attaches your polymer, okay, and then it exits. The empty tRNA moves out. So when there is a stop codon, the tRNA will not come. The ribosomes will move apart. So that is the end of your translation process. Alright, clear? So ribosomes are important players here. So cellular factory. So ribosomes are cellular factory which are responsible for synthesizing proteins. So what are ribosomes made of? Ribosomes are made of structural RNA, that is rRNA. So they are ribosomal RNA. So they have a larger subunit and a smaller subunit. When there is no translation happening, the larger and the smaller subunit remain separate. But when they encounter an mRNA, they attach together in the presence of Mg2+ ions. Okay. So small subunit encounters an mRNA and the process of translation begins. Done? Yes. Okay. So do I have this? Okay, I don't have the image. So it will be like this. Okay. So this is my mRNA 5', 3'. So this has poly A tail and this has guanosine methyl phosphate, and here I have my initiator codon. my stop codon, and this is my start codon and my stop codon. So my ribosomes will attach here and it will continue the process of translation till here, right? But in this region and in this region, I have some portion. So that is known as untranslated region. If you remember last class I told you, so here there is a small portion known as UTR. So similarly here there is a small portion known as UTR. So this is 3' UTR, this is 5' UTR. So UTR means untranslated region. So these two bits are not translated, but these regions are needed for the efficient process of translation. So that is what is given in the slides here. Alright, yes, okay. So inside your ribosome, inside the bigger subunit of ribosome, you have 3 places. This is E site, this is P site, and this is A site. So here I have my mRNA. So the first tRNA, which is carrying the amino acid, will come inside, okay, and it will have the amino acid here. The second tRNA will come and it will carry an amino acid. So once first tRNA comes and it has an amino acid, okay, so that is fixed. The second tRNA comes, then this tRNA moves out, so this goes here and this helps in the formation of peptide bond. The next one comes, adds the amino acid and goes out. So A site brings the amino acid, P site forms the peptide bond, and the E site exits the ribosome. So this is the E site, P site and the A site. Alright, so can you see this is the incoming one? So didn't you see in the GIF? So it was pretty clear in the GIF. So I showed you one GRF, right? Yes, this one. This one. See, this is forming your peptide bond. So this is the amino acid bringing tRNA. So here it forms the peptide bond and here the tRNA moves out. So this is the exit side. Alright, Antonia B, is this clear? Yes, Shubham sir. Hello, hi. A very happy good evening. So thank you for joining our lecture towards the end. So please do like, share and subscribe. So with this, translation process is done. I hope it's clear. So translation also has 3 stages: initiation, elongation. So initiation happens when it encounters your initiator codon, that is AUG, and it stops when it encounters your stop codon. Alright, is this clear? Yes. So translation is done. Yes? Okay, so in the next class, we will talk about the process of HGP and the regulation of human gene expression. So it is a very small process because I just have to explain lacopron to you and I have to give you the principles of HGP, and towards the end, we have DNA fingerprinting. So today is Wednesday, so I will finish the chapter on Friday at max. And yes, if you haven't understood A, P and E side, I will bring another GIF, so don't worry. Alright, yes, so please think about this crash course. This is once again a humble request. So please do join me for my motivational session. It is at 6 o'clock. So it is titled Good Things Take Time to Happen. So please join me for the same. And if you genuinely liked my way of teaching, so please do like, share and subscribe and join me for my 6 o'clock session as well. Okay? Yes? Chalo, bye. So take care, and this is me wishing you more and more. So see you at 6 p.m. Bye-bye.